on smart biomaterials for healthcare, organized as a part of the ANRF PAIR project of IMS University in association with the University of Hyderabad. May I now respectfully invite our esteemed dignitaries to kindly take their seats on the dais. Professor Dr. Sabu Thomas, Director, School of Nanoscience and Nanotechnology; Professor Dr. Radhakrishnan E.K., Professor, School of Bioscience and Principal Investigator, ANRF PAIR Project; Professor Dr. Bindu V.R., Director, School of Computer Science and Honorable Director of SARE; and Dr. Anas S. I invite Professor Dr. Gautam to be seated on the dais.
Please come. Yeah, please.
Now let us begin the inaugural session with a prayer. I invite Ms. Gayatri AP for the prayer. I request everyone to kindly rise.
നന്മയാകുന്ന കാന്തി കാണുവാൻ കണ്ണിനാകേണമേ നല്ല വാക്കിൻ്റെ ശീലു ചൊല്ലുവാൻ നാവിനാകേണമേ സ്നേഹമാകുന്ന ഗീതമോ എൻ്റെ കാതിനിണയാവണേ സത്യമെന്നുള്ള ശീലമോടെൻ്റെ ശാന്തി അറിയിക്കണേ ഭൂമിയമ്മയെന്നറിയുവാനുള്ള ബോധമുണ്ടാകണേ ജീവജാലങ്ങളാകെയും ജന്മബന്ധുവാകേണമേ ജാതിഭേദങ്ങളിന്ന ശാപമോ ദൂരെ മറയണമേ
Now I invite Professor Dr. Radhakrishnan E.K., Professor, School of Bioscience and Principal Investigator, ANRF PAIR Project, Mahatma Gandhi University, to deliver the welcome speech.
Good morning to all of you. Respected former Vice Chancellor Professor Sabu Thomas, dear Professor Mahesh Kumar, Professor Anas, Professor Bindu Madam, Dr. Gautam Chandra, and all the participants. So this is the first program that we are going to conduct, or we are initiating here on behalf of ANRF PAIR project. Unlike other project schemes, as you know that this is a particular model that Government of India is going to initiate or just started to implement across the country in such a way that more sort of interdisciplinary and collaborative research and translational ecosystem is to be developed. That is the major objective behind this scheme. And in this particular project, as we know that Mahatma Gandhi University is acting as a spoke institution along with the University of Hyderabad, which is a hub institution. And we took, along with other 6 institutions also, and all these institutions together received almost 112 crores of rupees 12 crores of rupees for this nationally prestigious program. And this was a highly competitive project selection, and out of the various projects submitted across the country, only 6 projects were selected for this much of funding, and that too among the various institutions of University of Hyderabad. Mahatma Gandhi University is again one of the well recognized and well reputed institution where much contributions are expected. That is the most important thing. And we were together for the kick-off meeting last week there at the University of Hyderabad. And there the message is that unlike other conventional projects and research work we are into, here it is for collaborative work and collaborative research, and where different sectors they have to interact and collaborate and work together for generating a different model. And we are coming here with a a different model from the university itself, where the project involves investigators from various departments, including School of Environmental Science, School of Chemistry, School of Nanoscience, and School of Computer Science, School of Bioscience, and also IUCBR. And these are the major institutions that are represented in this one. And here, all these investigators are there, and along with the School of Physics, Pure and Applied Physics also. So here, all the investigators are focusing mainly on diabetes and fatty liver in various aspects, ranging from development of sensors, ranging from drug development, and ranging from AI-based prediction and analysis on what is the social impact and societal impact of this type of disease that are there in the country. So that is at this particular platform that we are going to initiate such a collaborative ecosystem to build here. And here we have 9 project fellows working under 9 investigators, and we already had several meetings with the project fellows, and the project fellows are again suggested to have more interaction and collaboration with all the investigators here, and also they are supposed to work with investigators from the University of Hyderabad also. So this is a wonderful model that we are going to initiate, and it is a prestigious program of the country. And by the completion of this project, we expect NG University to get transformed into a hub institution where we may expect to receive a huge funding from central government for this type of projects to implement. This is the basic message with respect to how the project and what it means for a general understanding. So my responsibility is here to welcome all the participants, and first of all, I take this opportunity to welcome our senior mentor and former Vice Chancellor, Professor Sabu Thomas sir. And as we know, sir is a model for all the faculty members here, and the university has taken its highest level under the leadership of Professor Sabu Thomas sir. And we are actually blessed to have your presence, sir, and welcome you, sir, on behalf of ANR of Pair Project. And next, I welcome all our dear colleagues and our senior professor from School of Computer Science. I welcome Professor Bindumadan to this program. Actually, the entire project will be based on the support from our School of Chemical Science, because all the topics and research are focused on AI, and we had several discussions, and also University of Hyderabad is much more happy that we have a specific department also here on artificial intelligence and robotics, and so that is a good combination that our university is having uniquely. So welcome you, madam, to this particular program. Next is our director, School of Environmental Science, and Professor Mahesh Mohan. So I invite you, sir, to this particular program. And here we are again going to address one important and totally unexplored area, in the sense that how the environmental contaminants are directly or indirectly responsible for this type of disease incidents. So there the environmental science, now we are getting more connected to everything like AI and life science and everything. So welcome you, sir. And also the project, the government, the central government has already identified the theme as MIC, metabolic disease, infectious disease and cancer. This is the theme. And Mahatma Gandhi University will be addressing metabolic disease only. and that is our major theme. So in that point of view, like my sir will also focus on conducting large number of field survey and how the disease is there across our we welcome our DCDC and the Director School of Pure and Applied Physics, which are also to come over the. Please. Yeah, so maybe it's a massive samples we will be analyzed, and for understanding how these disease incidents are there with respect to different sectors of society, and what are the environmental factors responsible, and if so, what sort of predictions that we can make on to predict the incidence of disease and also to control how it happens. And now I welcome our Director School of Pure and Applied Physics and Senior Professor DCDC, also Directorate of College Development Council, Professor Biju sir. Sir, welcome sir to this particular program. And this is also very important context that sir is working on biosensor development. So the sir has already published one paper related to that one. So here the objective, the project work sir is taking care of is on development of sensors or biosensors for the rapid, cost-effective diagnosis of this disease. That is also very important as we are moving ahead with the Vikasit Bharat 2047. Our major mission is to develop more sort of cost-effective, rapid, and non-invasive devices and methods. And sir is working on that one. And welcome you, sir. And next is our professor from School of Chemical Science, Dr. Anna sir. Welcome you, sir, to this particular program. Sir is working on another very important area that, as we know that there are various types of chemicals or drugs that we are used to manage all these conditions. And sir is working on how novel drugs can be developed by changing its particular chemical nature or functional group or structure and those things. And that is also very important area where sir will be addressing how the existing drug-based or natural product-based or novel drug combinations can be developed. That is also very essential because as we know the biopharma industry is a major sector where we need to have more focus on developing indigenous drugs and medicines. That is the area sir's expertise will contribute. And next I welcome Dr. Gautam Chandra, Senior Scientist from Inter-University Center for Biomedical Research. Welcome, sir, to this particular program. And IUCBR will be working mainly on, again, on the clinical and biochemical basis of diabetes and fatty liver. So these are the major areas. As you might have visited that IUCBR is having a wonderful facility, and it is having the animal house, which is second to Sri Renal Institute, and it is the second biggest or animal house facility the state is having. And why I mentioned all these things are because we are going to develop a translational research ecosystem where irrespective of the subject, irrespective of the area, the students, researchers and the faculty members are expected to interact with each other. Like we have to get transformed into a collaborative and a mission mode approach. That is the word that ANRF also introduced to us. It is a mission mode approach. So much changes and transformation should happen. And also I welcome all the participants to this particular program. And because of your presence only, this program is happening. And also this program is, we expect that this will going to make some significant impact on your research aptitude and also to identify the aims and objectives of your project. Welcome you all, and I also take this opportunity to welcome our respected resource person, Dr. Prashant Sadanandan sir from Government Medical College, Kottayam. Welcome you, sir. And on behalf of Mahatma Gandhi University, we welcome you to this particular program, and also we welcome you to be part of our project initiative. As it is already mentioned that we are working in academic background only. We don't have any clinical expertise or background, and this project that we are addressing is a clinical issue, and where without a medical support the project will not be successful. So we already had 2 rounds of meeting with the Government Medical College, and Puno sir, principal, was very much supportive, and one meeting conducted here and second meeting was there, and we already identified several teams and groups And we also request your support for the success of this program, and I welcome you all. Thank you very much.
Thank you, sir, for your warm and thoughtful words. We now move to ceremonial lamp lighting, symbolizing the light of knowledge. May I request the dignitaries on the dais to kindly come forward for the lamp lighting ceremony.
Please come to the dock also. Please. Yeah. You are our first speaker. Are we all positioned well?
I think this moves
Forward, yeah. Okay? Yeah. Thank you. And with all your permission, I declare that the meeting is open. Thank you. Yeah, you are our senior most professor, lady professor, yes.
This year science day.
Yes, yes, yes. Excellent.
Thank you. Please take your seats. It is now my privilege to invite Professor Dr. Sabu Thomas, Director, School of Nanoscience and Nanotechnology, Mahatma Gandhi University, to deliver the inaugural address.
Very good morning to all of you. Professor Bindu, Dr. Anas, Professor Biju, Professor Mahesh Mohan, Professor Radhakrishnan, Professor Gautam. Our first speaker today, Dr. Prashant Sadanandan from the Government Medical College, Kottayam. All my colleagues, postdocs, faculty members, students and participants. If you look at this stage, you will find that the stage is an example for interdisciplinary, multidisciplinary research. We have really physicists, chemists, AI, Professor Bindu actually heading the AI department, biologist, chemist, nano people. So it's really a nice stage where people from different domains work together. Probably you all know that Mahatma Gandhi University has won the PAIR program, Partnership for Accelerated Innovation Research. A new program introduced by the Government of India, NRF, to accelerate research, working with the different institutions. And because of our higher ranking, Mahatma Gandhi University could participate in the competition. And you all know that our hub, top-tier institute is University of Hyderabad, one of the leading universities in the country. And we have now 6 or 7, how many, Radhakrishnan? nine, spoke institutions. And Mahatma Gandhi University won this, and we are going to get really good funding. As you all know, the topic is very interesting. It's more on the diabetic and fatty liver, and the program is headed by Professor Radhakrishnan, our colleague. And today, as part of the PAIR program, we are organizing a 2-day national seminar. I really like the title of this topic, Smart Biomaterials for Healthcare. Very important topic. We all know that our country is the most massive country in the world. We have beaten China, very close to 1.4 billion people. So healthcare is extremely important. And today we are going to have a very exciting meeting on smart materials, smart biomaterials. Smart biomaterials are intelligent materials. They respond to different types of external stimuli. Smart materials can respond to temperature. Smart materials can respond to pressure. Smart materials can respond to magnetic field. Smart materials can respond to pH, and their conformation changes with respect to external environment. And there are lots of applications in drug delivery, in medicine, for the manufacturing of scaffold, dynamic implants, lots of applications. And there are fantastic materials being developed by different companies now. If you look at the market of smart materials, there are many companies that are active from Europe and United States making smart materials for a variety of applications. And dear friends, today we have 4 speakers. The first speaker is Professor Sadanandan, and then we have Professor Denny Joseph. Both are from Government Medical College, Kottayam. We are going to have active cooperation with them. So whatever we develop at the academic institute should reach the masses. That is why the participation of our doctors is extremely important. And 2 more are from our own former co-workers. Dr. Vinod was from chemical science, and he is now heading one of the important units in Rajiv Gandhi Institute of Biotechnology. And Professor Gia worked with me for about 5 to 6 years on bio-nanomaterials. So we have 4 speakers. So I request all my students, faculty members, listen to all the talks, understand the latest advancements in the subject of smart biomaterials. And that will be extremely useful to continue the work in a very successful manner. At the end, we should make quite nice devices, for our people. That is the end use of this accelerated partnership program. So I wish all the best for the program. With all your permission, I declare that the 2-day national seminar on smart biomaterials for healthcare has been inaugurated. And I wish all the best of this success. I congratulate Radhakrishnan and Dr. Sreekala in organizing a very beautiful meeting. Our Vice Chancellor could not come today because he has some very important meeting. That is why I was asked to open this meeting. Thank you so much.
Thank you, sir, for the inaugural address and your valuable presence. Once again, I thank all the dignitaries on the dais for their valuable presence in making this session a memorable one. Thank you. As we are running short of time, we are formally concluding our inaugural session. Now, I invite my colleague Gladys to propose the vote of thanks.
A very fine good morning to one and all present here. Respected dignitaries on and off the dais, esteemed speakers, faculty members, dear research scholars, students, everybody, good morning. So it's my proud privilege to propose the vote of thanks for this inaugural function of this 2-day national seminar on Smart Biomaterials for Healthcare, organized by the School of Nanoscience and Nanotechnology as a part of Science Day 2026 under the ANRF PAIR initiative in collaboration with MG University and University of Hyderabad. First and foremost, I express my heartfelt gratitude to Professor Dr. Sabu Thomas, Director, School of Nanoscience and Nanotechnology, for his inspiring inaugural address and his constant encouragement and support towards the research and innovation at Mahatma Gandhi University and our School of Nanoscience and Nanotechnology. My heartfelt appreciation also goes to Dr. Radhakrishnan E.K., Principal Investigator of the ANRF PAIR project scheme of the MG University, for his warm welcome and for his dedicated steering in this prestigious project forward. So I would also like to thank Dr. Sreekala MS in her absence to acknowledge her constant support to guide the ANRF PAIR project under the Department of Nanoscience and Nanotechnology. And also I congratulate her for her dedicated efforts in organizing this two-day national seminar. I also extend our sincere thanks to all the dignitaries on the dais, Dr. Gaudam, Dr. Bindu Veer, Dr. Sabu Thomas, Dr. Anas, Dr. Mahesh, and Dr. Biju for their valuable presence and their time. Finally, I would like to thank all the participants and delegates for your enthusiastic presence and active engagement. So with sincere gratitude to everyone contributed directly and indirectly to the success of this inaugural function, I conclude my words. Thank you.
Thank you, Gladys. With this, we come to the end of the inaugural session. The tea is arranged outside, so I invite all the dignitaries to join us for tea. Sorry for the inconvenience. Now we are going to start our technical sessions. It is my privilege to introduce Dr. Prashant Sadanandan, Assistant Professor at Government Medical College, Kottayam, under the Kerala University of Health Sciences. Dr. Prashant holds a PhD in pharmacy and has extensive research experience at the interface of pharmaceutical chemistry and biology. His works primarily focus on nanotechnology-based drug delivery systems, particularly in the treatment of autoimmune diseases, cancer, and neurodegenerative disorders. He has received several prestigious national and international awards and has contributed significantly through high-impact publications and patents in the field of nanomedicine. We are honored to have him here today to share his insights and expertise.
Hello. So, a very good morning to all of you. So, first of all, I take this opportunity to thank the organizers of this 2-day seminar, Smart Biomaterials for the healthcare sector. And thanks for the organization for giving me an opportunity to present my work here. This presentation is entitled as Nanobiomaterials for Immunomodulations and its therapeutic applications. So, before I get into those, the actual research work, I will first 10 to 15 minutes I will take you to the basics of immune system, nano-related works, which already you might be knowing. Then I will get into the area where I am working. Hope I am audible to the backside. Okay. So I work basically at the interface of nanotechnology and immunology. So that's why I chose this topic for the presentation. So you know, in the case of immune system, they are basically the immune system is to attack against the foreign microorganisms, or keep a balance in our body, or to have a balance between the pro-inflammatory cytokines and anti-inflammatory cytokines. And these components, like you know that B cells, T cells, as a part of adaptive immune system, whereas the macrophages, dendritic cells, NK cells, natural killer cells, they are part of the innate immune system. And in the immune system, or this immunomodulation, one of the important part is managed by the lymphoid organs. As you know, the primary lymphoid organs are the bone marrow and thymus, and the secondary lymphoid organs like spleen, lymph node, and the Peyer's patch plays a huge role in immunomodulation, whether it's cancer or the other condition, opposite condition like autoimmunity. This lymphoid organ plays a huge component. Now, the lymphoid organs mainly mediated by this B cells, T cells. I will come into those things. Now, this immune system, we can classify, you know, the basics. We can have it into two types: innate system and adaptive immune system. The innate system basically the inborn immune system. Okay, so they basically contains this macrophages, neutrophils, NK, natural killer cells, dendritic cells. So they basically recognize this foreign things, foreign organism based on this pathogen associated molecular patterns, or the pattern recognition receptors. So once it is activated, this natural killer cells or this dendritic cells will get activated. That will further activate the system and start attacking these microorganisms. Now, when it comes to adaptive immune system, that is the most important part, where the system contains mainly the T cells and the B cells. T cells are basically CD4 T cells, that are helper T cells, and the other one is the cytotoxic cells called the CD8 positive cells. And to regulate all these things in a right way, or to keep a balance, we have this regulatory T cells, which plays an important role in immunoregulation. Now, B cells, basically the humoral immunity, they produce antibodies. You all know those things. Now, the cell mediated, the cells CD4 and CD8 can be included in the cell mediated immunity. Now, this adaptive immunity creates this memory. That's why whenever we encounter one disease, and we have a resistance towards it in the future, actually. Now, why this immunomodulation is required, actually, or why you need to work in this immunomodulation condition? So, the primary area, or the most of the immunomodulatory studies are now focused on the cancer, because you know that those days where the small molecules which can go and inhibit those cancer cells are gone. Now it's a time for cell therapy, it's a time for immunomodulatory therapy. That's why you look into the Nobel Prize winners in the recent things. They are all not for this small molecules. They are mainly focused on the latest advancements, where you are focused on immunomodulatory stuff, or cell therapy or stem cell therapy or other things. Now the other part, immunomodulation is very important in autoimmune disease, which is in the past decades that is not much focused, but recently a huge works are carrying out on autoimmunity, because again the basic treatment for autoimmune disease was immunosuppressants, but it is not specific. So a lot of other toxicity can come, and it can result in various side effects. So now immunomodulation is also important in autoimmune disease, and other allergic reactions or inflammatory conditions. These are all rising, and here also a lot of immunomodulatory works are going on. Now, you look at this conventional therapies where I talked about small molecules or the basic non-targeting, they have a lot of side effects. They are not specifically treat or regulate those particular disease. And due to this, they have a lot of side effects. For example, if you take autoimmune disease, the basic treatment is immunosuppressants. Now, it is not specific. Due to that non-specificity, the patient will undergo various other side effects, or the patient will be prone to various side infections. So that is the toxicity related, or the bioavailability is an issue. Your targeting will be less, or the amount of drug reaching that particular target will be very less. So that is all limiting or the creating the efficacy, or reducing the efficacy and creating toxicity. Now there comes the role of nano, where you can precisely target the diseased tissue, or you can precisely target those areas where you wanted to deliver the drug. You can control the delivery by kinetics. You can have a sustained release, slowly releasing the drug at the particular point. Also, you can reduce the dose of the drug by incorporating a nano carrier. You can reduce those doses, and which can further reduce the side effects. So there comes the role of nanotechnology. Now, I don't need to teach you about nano, because you're all coming from the background of nanoscience department. Now, just this slide I've included to put a comparison between the atomic scale and the macro scale. Now, you look at this amino acid tryptophan and having a size of 1 nanometer, or you look at the bacterium or having more than 1 micrometer up to 10 micrometer. But we are dealing with something very small, tiny particles coming in the range of 1 to 100. That is a normal definition what is given for nano. However, up to 1000 we still consider it as a nano, larger nanoparticles. So normally nano in the sense, or nanoparticle in the sense, it ranges from 1 to 1000 nanometer. Okay, so we are dealing in such small level, and due to that small level, a lot of advantages are there, which will come to in the next slides. Now, what are nanobiomaterials? So basically, we can split this term into three: nano, bio, and materials. Nano, I have already told, nano means very small stuffs. We are dealing with 1 to 1000 nanometer size. Bio in the sense, it should be biocompatible, or a biological material, or a biodegradable material. So those things comes under bio. And materials could be anything. It can be lipids, or it can be carbon, or it can be inorganic particles, polymeric nanoparticles. It could be anything. So that comes, or this is that actual definition. We no need to look into the definition, but they are something which is biocompatible, having a very small size, and which can be used for therapy, diagnosis, which is combined called as theranostic. So we can include in various applications. Now, the advantage would be, when it comes to a nano level, the surface area to volume ratio will be very high, and due to this thing, the interaction towards the cells will be huge compared to a bigger molecule. Now, again, we can really engineer these nanobiomaterials into different sizes, different compositions, and we can adjust the porosity so that we can load the drugs into a higher quantity also based on those porosity. Now, due to this nano effect, we can, especially in this tumor case, you have heard about this enhanced permeability and EPR effect. We can passively target this tumor tissue. Also, as Sabudana sir was telling in this introduction, we can just respond or make this nanoparticle respond to various stimuli. For example, it can be adjusted or engineered to change or modify with respect to pH or temperature or just certain stimulus. So we have all the control to regulate that, and there comes the trick, or there comes the engineering of nanobiomaterials, which is very important in regulating various disorders. Now, classification, there are a lot of ways to classify based on the size, based on the shape and all those things, and this is based on this composition. Just taking you to this organic compound nanoparticles, inorganic, biomimetic. Organic in the sense, lipid based particles are there, the liposomes. The most widely approved nanomedicines are basically liposomes actually. Now, polymeric particles are there. PLG is FDA approved material. You have this natural material called as chitosan. Dendrimers are there. Nowadays, you know that the protein itself, the human blood contains albumin, globulin protein, right? This albumin is taken out from the blood and they are converted to nanoparticle. That means it biomimics, or it is biocompatible to our system, and that is actually that has come as a drug also. There is a drug called as Abraxane, which is nothing but albumin bound paclitaxel. Paclitaxel is a chemo drug. So even our proteins are converted to nanoparticles, which is a very, very good thing to have this biocompatibility. Now, whereas you know that vaccines, the COVID vaccines, mRNA vaccines, and so all those are lipid formulations and of mRNA vaccines. So they are used. Now coming to inorganic metallic nanoparticles, very long before itself it was used gold, silver, platinum, even iron oxide is used for both treatment as well as diagnosis. There is a theranostic purpose. You can use it for imaging magnetic nanoparticles. There are silica-based nanoparticles which are very much used. Nowadays, recently, most of the work is going on this kind of biomimetic materials, like you are using the cell membrane to coat over the nanoparticle so that it will mimic to our body's own cells or the material. So those kind of cell membrane coated nanoparticles are used. Metal organic frameworks are coming, and a lot of other works are going on this thing. Now this is classification based on dimensions, the porosity. As I told you, we can engineer the nanoparticles at different pores so that you can load the drugs of different sizes. Load, for example, you can add an amino acid, you can bring a protein into that based on the porosity. So based on the porosity, nanoparticles can be classified into microporous, mesoporous, macroporous, and all that. Now, as I already told you, some of the nanoparticles are basically natural, chitosan. They are of natural, and like this we can classify nanoparticles. Now this is again a pictorial representation of various nanobiomaterials. I will just skip it, all these things. Now this part which I really wanted to focus. Now immunomodulation, that is the topic of my talk. Immunomodulation could be of two types. You can stimulate the immune system, that is immunostimulation, or you can downregulate or you can depress that immune system, that is called as immunosuppression. Now why this, where, in which context you need to stimulate, in which context you need to suppress the immune system. Now if you look at immunostimulation, you look at cancer, as one Nobel laureate has told, whatever discovery we do against cancer, it's like a chess game. Whatever movement we make, the cancer will make a counter move. Okay. Now that's why I told you those days where the small molecules or the cytotoxic drugs has gone. Now it's the rise of cell-mediated therapy or immunotherapy. Now in this cancer, we need to kill the cancer cells, so we can stimulate our own immune system like T cells, B cells, and activate that cells and target or make it attack those cancer cells. So you need an immunostimulation in the context of cancer. You look at infectious disease. There also you wanted to stimulate the immune system and make it attack towards the bacteria or virus. You look at the vaccines, COVID vaccines, we need to, or any vaccines, they will stimulate the immune system and attack those microorganisms. So those are the contexts where you need to stimulate the immune system, cancer and all those things. Now in which context you need to depress that immune system or downregulate the immune system, there comes this autoimmune disease. Now in autoimmune disease, I will tell it in detail. Our immune system is already activated and they are already attacking our own body cells. So in those cases, we need to downregulate that immune system, that is immunosuppression, so we can utilize in this autoimmune disease. Now another area, that organ transplantation, you might have heard of kidney transplant, liver transplant. So whenever an organ from a different person is put it in our body, the immune system will be activated. They will try to reject it at any context. So in that context, whenever an organ transplant is done, the medications which will be given will be immunosuppressant, because the immune system has to be depressed. Then only that organ can stay there for some time, for a longer time. Now, in those cases, transplantation, especially organ transplantation, you need to downregulate the immune system. Now, you look at allergy, where the immune system is very much activated. There also you need to downregulate the immune system. So these are the different contexts where you need to think where you need to stimulate the immune system or where you need to depress the immune system. Now, the ways to stimulate the immune system, one way is, a lot of strategies are there, just I'm taking the important things. So one is that we have the cytokines called as pro-inflammatory cytokines, which will activate the immune system. So those cytokines we can activate, pro-inflammatory cytokines like IL-2, interleukin 6, TNF-alpha. That is one way to activate the immune system. The other way is that we need to activate this pattern recognition receptors. Like we need to give some adjuvants along with our drug, so that will activate those pattern recognition receptors that will further complement and activate our immune system, T cells and B cells. They will further go and attack those cancer cells and all that. Now the another thing is that this is a brake system that is called as the PDL1, programmed death actually. So this is a kind of a brake actually. We need to inhibit those brakes. then the immune system will be super activated and it can go and go. So one of the 2022 the Nobel Prize was awarded for discovering this PDL1 inhibitor actually. Okay, because when you are inhibiting this pathway, it can activate the immune system and that can further go and attack the cancer cells. So that was the discovery which has won the Nobel Prize actually. Another way is that you know that normally the immune cells will present the pathogens or the bacteria towards this T cells or B cells. That is done through this MHC1, major histocompatibility complex. So you can activate those presenting part also so that the immune system will be activated. Now coming to this immunosuppression, it's just the opposite. Now here I told you pro-inflammatory cytokines, but when you need to depress the system, this anti-inflammatory cytokine, basically this should be in a balance, this is normally in a balance in our body. When the balance is lost, it may end up in a cancer or can end up in an allergy or an autoimmune disease. So these are the anti-inflammatory cytokines which is normally focused to downregulate the immune system. One is the interleukin 10, then the TGF-beta and all that. Another way is that earlier I told you need to inhibit this program death ligands, but here you need to activate this ligands so that will bring the immune system down. Now this is a very important part, regulatory T cells, which regulatory T cells are just like a patrolling, just like a police who will make sure that the law and order is obeyed. So like that, regulatory T cells plays an important role in maintaining or balancing the immune system. So increasing or activating this regulatory T cells is a very good way to downregulate the immune system. And recent Nobel Prize was won for the discovery on regulatory T cells. I will come to those slides. Now the various applications I have already told you, cancer immunotherapy by activating the immune system. Now in the autoimmune disease, by downregulating the immune system, you take any autoimmune disease, rheumatoid arthritis or multiple sclerosis, the normal treatment is immunosuppressants, which has a lot of side effects. Now the other way is that specifically downregulate the immune system, which is again a immunosuppressive technique actually. Now apart from this, the infectious disease, as I told you, COVID vaccine, Moderna vaccine, BioNTech vaccine, they are actually a part of nano-immunomodulation which has come. We have all used actually BioNTech and Moderna vaccine. Now apart from that, adjuvants, vaccine adjuvants also, there also this nano-immunomodulatory or biomaterials plays a huge role actually. Now coming to this important, what are the nanomedicines that is available in the market? Currently more than 100 nanomedicines are in the market, FDA approved drug. But major, I will just take you to the major one. The first one was Doxil. It is basically a liposomal based formulation. Doxorubicin is an anti-cancer drug. So this was the first FDA approved anti-cancer nanomedicine, liposome, which has come in 1995. Then various other immunosuppressants with the nano part has come. So these were the important key nanomedicines by various, even you look at this COVID-19 vaccines, I told you, Moderna vaccines and all those things. Now various inorganic nanoparticles are coming into the play for immunomodulatory things. Now if you look at the various nanomedicines, major nanomedicines which is in the market are more than 53% are basically for cancer treatment. More than 53% nanomedicines that is approved in the market is mainly for the indication of cancer. The remaining all covers the rest of the percentage. Okay, so there indicates the importance of nanomedicine in immunostimulating the stuff for regulating the cancer. Now, if you look at this approved cancer nanomedicine, as I told you, Doxil was the first approved nanomedicine. Later on, a lot of drugs incorporated with this different kind of nano formulation has come into the market. Most of thing, if you look at here, liposome, it's mostly the liposome actually. So if you look at this nanomedicine formulation, more than 33% are liposomal formulation. Liposome is basically lipid bilayer containing nanoparticles. So it has a hydrophobic shell and a hydrophilic core actually. So that is the mostly FDA approved thing because it's mostly biocompatible to our body, and liposomal works are most of the formulations are basically liposomes actually. That doesn't mean other formulations are not there. There are even inorganic nanoparticles, other protein-based nanoparticles, everything is there. But just to indicate the importance of liposomes, I just put the slide. Now with that small intro, I will just get into where I am working. So basically I am working at the interface of nanotechnology and immunology. So that, as I told you, immunomodulation. We can activate the immune system or suppress the immune system. So that is the area where I am working. Now, before I get into that, can you just, I would start off acknowledging this personalities. Can you recognize for what they have recognized, or why we need to acknowledge that? Mary Branco, Fred Runsell, and Shimon Sakaguchi. So they recently won the Nobel Prize in 2025 in Physiology or Medicine. And for what discovery they have awarded Nobel Prize? For this phenomenon is called as tolerance, or earlier I told you the discovery of regulatory T cells. This is very much important to tolerate or keep our immune system into a tolerance fashion. So we can in the healthcare sector, this tolerance, I will explain what is tolerance and all that. In healthcare sector, two important areas where this tolerance is important is, one is autoimmune disease. The second is organ transplantation. In both the condition, you need a tolerance because your immune system is already activated, super activated, and it is attacking our own body cells. So you need to create this tolerance. And for those discoveries, they have been awarded Nobel Prize. Now, I work in this area. I told you two important health sectors where tolerance can be applied is, one is autoimmunity, the second one is organ transplantation. So I basically work in autoimmunity. So you know that our immune system is supposed to attack foreign organism or foreign microorganism or whatever thing, or protect ourselves from outside things. Now, in autoimmune case, our immune system, or there will be a dysregulation, or there will be failure of immune regulation, and our immune system start attacking our own cells, our own body cells. So that is called as autoimmune disease. And Kerala is a hub of type 1 diabetes mellitus, rheumatoid arthritis. They are all autoimmune disease. Other autoimmune diseases include multiple sclerosis. Even psoriasis has this autoimmune component, systemic lupus erythematosus. So this autoimmune disease is basically occurring due to the failure of immune regulation, and our immune system start attacking our own body cells. In type 1 diabetes, what happens? Our immune system start attacking beta cells, which produce that insulin. That's why the glucose cannot be controlled. In rheumatoid arthritis, what happens? The immune system start attacking the joint cells, the cartilage or the proteins in the joints. That's why rheumatoid arthritis patients will have stiffness or difficulty in walking or joint pain and all, or inflammation comes. Now, we started off, my work started off by taking this autoimmune model called as multiple sclerosis, where there will be a failure of immune regulation and our immune system attacks the myelin sheath. You know that spinal cord has this myelin sheath which helps in the conduction of impulse. Okay. Now this immune system start attacking the myelin sheath, and the patient will not be the conduction of the impulse in the patient will not happen. The patient starts showing the symptoms and finally end up in wheelchair. Okay, walking disability or memory problems, and finally end up in bed actually. So it's a silent killer. So they mainly attack this myelin sheath, the proteins present in the myelin sheath. Our immune system start attacking. Now, some of this proteins present in the myelin sheath are myelin basic protein, myelin oligodendrocyte glycoprotein, and this PLP called. So these are the proteins present in the myelin sheath, and now this immune system start attacking these proteins, and that will cause the neurodegeneration in that patient. Okay. Now, MS, although initially this was very common in the Western countries, now the MS is on rise in India, especially young adults, especially female young adults at the age of 18 to 25. There are a lot of patients are coming to the clinic. Okay, earlier institute where I was working with at Amrita, every week in the neurology department, 3 to 4 cases was coming. That too young adults, and okay, with MS. So nowadays it's on rise in India. Now, you take any autoimmune disease, as I told you, rheumatoid arthritis or type 1 diabetes mellitus or multiple sclerosis, the primary treatments are immunosuppressants. So these drugs, they go and downregulate the immune system, they non-specifically suppress the immune system. Now in the case of MS, these are also immunosuppressants, they go and downregulate the immune system in a non-specific way. Now our immune system is completely suppressed. What will happen? We are prone to other infections when the immune system is completely suppressed. So the patient will end up in various other infections, and that will create other problems. So that is a disadvantage of immune suppression, and that is the primary treatment. In fact, we don't have any other solution for that. So normally immunosuppressants are given. Now, this was the problem. So we thought of going with a different approach. as I told, to induce this phenomenon called as tolerance. So I have been working for the last 8 years in inducing this phenomenon called as tolerance by using these nanomaterials. So advantage of this thing is that it can specifically modify this autoreactive T cells, because in MS it's the autoreactive T cells mainly they go and attack our own cells. Now, so we can specifically modify those autoreactive cells, and it will be specific compared to an immunosuppressant which non-specifically downregulate all the immune cells. So here we can specifically regulate this autoreactive cell, and we can induce this phenomenon called as tolerance. And that's what I told earlier. The recent Nobel Prize was awarded to this particular discovery, inducing the phenomenon called as tolerance. Now, what exactly is this tolerance? So if you look at the definition, by definition it says that suppression of an immune response to a particular antigen by administrating the same antigen. So that is you suppress the cellular response, you suppress the B cell response, humoral response by administering the same antigen, and that will suppress the immune response to that particular. I will just explain it in a different context. We humans take lot of food antigens. Whether you are a vegetarian or a non-vegetarian, we all take a lot of food antigens. However, fortunately, we do not end up in an autoimmune disease. The reason is that we have a tolerance-inducing mechanism, natural tolerance-inducing mechanism in our body, which is mainly mediated by this tolerogenic lymphoid organs. As I already told you, spleen, lymph node, Peyer's patch, those lymphoid organs plays a huge role in managing or maintaining this tolerance. That's why, although we take a lot of food, even non-vegetarians sometimes take mutton liver, mutton brain, mutton liver, or even in vegetarian they take sometimes peanut, sometimes it will be better. But still, most of the times we do not end up in a autoimmunity. The reason is that we have a tolerance-inducing mechanism, and it's mainly mediated by the this lymphoid organs like spleen, lymph node, or the Peyer's patch and all that. So similarly, what we can do is that if we can administer those autoantigens, which are the culprits of or the targets of this autoimmune mediated attack, to these lymphoid organs, if we can deliver this autoantigens to these lymphoid organs, we can modify the immune response into a tolerance fashion. Okay, for example, in multiple sclerosis, I told you the immune system start attacking the myelin antigens. So if you can deliver those myelin antigens into those tolerogenic lymphoid organ, we can engineer or modify the immune system into a tolerance fashion and suppress the disease to that particular autoantigen. So that is called as immune tolerance. Now, people have, researchers have tried administering these autoantigens either through the intravenous route or through the oral route. Now, the problem is that when you are delivering this autoantigen, because they are basically proteins or peptides, so when you are administering either through IV or oral route, most of it would be degraded within the circulatory system, or if you go through the oral route, in the GIT it will be degraded. So before reaching these lymphoid organs, it will be degraded. So that is one of the limitation when you are administering the bare autoantigen as such. Now, one way to address this thing is that deliver this autoantigen in the context of cellular carriers. Now, you know this apoptotic cells or phagocytic cells, or you take the splenocytes, and now you add or you dope or you conjugate this autoantigens onto the cellular carrier, then you try to deliver it. Now, the good thing is that when it is apoptotic cells or phagocytic cells or cellular carrier, they will be readily taken up by the macrophages, and macrophages will take it to the lymphoid organs. So this would definitely increase the bioavailability of this autoantigens in these lymphoid organs. So that is the area cell therapy, what has been already going on since a long decade. Now, the advantage of the cellular carrier is that they can deliver this autoantigens specifically to these tolerogenic lymphoid, because tolerogenic lymphoid organs rapidly uptake these apoptotic cells or phagocytic cells. Now, these are some of the reported methods or which have used cellular carriers, and they are shown to be highly efficacious in various clinical trials, including the multiple sclerosis patient. So they used this apoptotic cells, especially splenic cells. They conjugate with autoantigen, deliver it, and they have shown to be safe and efficacious in this clinical trial. Now, they have demonstrated that this apoptotic cells or cellular carrier will be readily taken up by the tolerogenic lymphoid organs like spleen, especially the macrophages in the spleen called as Marco, that would increase the bioavailability of autoantigens, and this would modify the immune system into a tolerance fashion. Okay, when you give bare autoantigen, it will not reach, but when you deliver it in the context of apoptotic cells, the bioavailability will be very high. Now, the problem when you are dealing with cellular carrier is that you need to isolate the cell from the patient. From the bedside, you need to isolate, and managing this safety conditions or this from the bedside back to this bench side, you need to formulate those things. So all this need a strict aseptic condition. It's very tough to manage those aseptic conditions when you are dealing with cellular carriers, because you have to get from the bedside, then you have to go to the lab, you have to do that and get back to the patient again. So strict aseptic condition is needed for the cellular carriers. So that is one limitation. Now the other thing is that the cost of production, when you are isolating a cell from the patient, then doing the chemistry, then administrating back to the patient, it's a long process. Again, it's a cost of production. Now the regulatory constraints will be a big problem. All this limitations will question the translational feasibility of using cellular carrier. Now we ask this question, instead of the cellular carrier, can we come up an alternative for this apoptotic cells or cellular carrier? So as you all know, in the case of nano drug delivery using nanoparticle, what is the major limitation? Suppose you wanted to target the brain, or suppose you wanted to target the pancreas. What is the major limitation when you are delivering a drug through in the context of nanoparticle? Immediately the nanoparticle, most of the time the nanoparticle will be taken up by the phagocytic system, mononuclear phagocytic system, or the macrophages or the dendritic cells. So then the drug will not be able to reach the targets. So that is one of the major limitations in nano formulation, the phagocytic uptake of nanoparticles by this phagocytic system, mononuclear phagocytic system. So this hinders the successful nanotherapy, correct? Now, so that is a normal case. Now we ask this question. In our case, especially in autoimmune disease or intolerance condition, we need to deliver this autoantigen to these macrophages or these lymphoid organs. So we ask this question, can we come up with an nanoparticle which will be readily taken up by these macrophages of the lymphoid organs? Okay. In the case of normal drug delivery or cancer therapy, this is a limitation. Uptake by the macrophages, uptake by the phagocytic system is a limitation. But in this autoimmune condition, we wanted to deliver it to the macrophages, or we wanted to deliver it to the lymphoid organ. So that is an advantage, or we converted that limitation into an advantage. So we thought of, can we come up with a nanoparticle system which can replace the cellular carrier, so that, and which can be readily taken up by these lymphoid organ macrophages. Now we played with a lot of nanoparticles. So before that, so our whole strategy was to develop a nanoparticle system that will be readily taken up by these tolerogenic lymphoid organs like spleen or lymph node or Peyer's patches. That was our whole strategy. So we started off with a lot of So I was telling, our plan was to develop a nanoparticle which will be readily taken up by the macro. So we played with a lot of nanoparticles. Sorry for the interruption. So we initially worked with a lot of nanoparticles like polymeric nanoparticles, carbon nanoparticles, then calcium nanoparticles, inorganic nanoparticles, and out of that we found that silica is a widely explored nanoparticle, and that has got a specific affinity towards this lymph node organ, especially in the spleen. Okay, so this is the near infrared image. We injected this fluorescent silica nanoparticle to the mouse, and we took out these organs. And actually, we did an live in vivo imaging. We could find that these particles are lodging into this abdominal area. We took out this organ, and this is the untreated control, and this is a treated control. You look at this is a spleen. It becomes white. That means that is the intensity, maximum intensity is for white. So that particle is getting accumulated towards that spleen. So out of various nanoparticles, we have got the best result with silica. So we took this silica as the nanoparticle to deliver this autoantigens. Okay, so that was the rationale, or that was the initial cue we got it from our study. Now, so we to get that, we took the silica nanoparticle of an approximate size 200 nanometer. So this was the carrier system for delivering this autoantigens. Now, as I told, the autoimmune model what we have used for this study was multiple sclerosis. And you know that in multiple sclerosis, our immune system attacks the autoantigens in the myelin, and one of the main autoantigen is this protein called as MOG, myelin oligodendrocyte glycoprotein. Now, to test this concept in the preclinical model, we took out this gold standard model of multiple sclerosis called as the EA model, where this EA model will mimic almost the same symptoms of this human multiple sclerosis. So that was the model what we used for the study. Now we engineered these particles. This is the normal functional groups which normally comes out of the silica. We engineered it. We introduced a lot of amine groups so that will increase the affinity towards these lymph node organs. We later on loaded these autoantigens to that. This was the method what we used. We used basically a sol-gel method where silica precursors were hydrolyzed and condensed to get these particles. Now we'll get into those data. So this is the DLS data where you can see this uniform distribution and a polydispersity index of 0.05 that indicates that particles are monodispersed, they are not clumped, aggregated. Now this is the electron microscopy image, scanning electron microscopy, and this is the transmission electron microscopy. So you can see that the synthesized particles was indeed spherical in shape, or 200 nanometer approximate size, and monodispersed also, both electron scanning as well as transmission. We confirmed it using the EDAX analysis where we got the peaks for silicon and oxygen. Now, once the particle is synthesized, we conjugated the autoantigen called as the MOG onto the surface by using this covalent bonding carbodiamide mediated chemistry. However, the loading efficiency, the loading of this protein MOG into this nanoparticle was very less actually. But we further did all the studies with this loading efficiency, approximately 21%. I will come to those data. Now, our aim was to see whether this particle is actually getting into those macrophages in the lymph node organ. That means we wanted to image, we want to develop an imageable nanoparticle. So for that, what we did is that we doped this iron oxide, which is a contrast agent, onto the surface of this particle. You look at this is the electron microscopy of silica, and these gritty particles are nothing but super paramagnetic iron oxide. So this will help you to image using MRI. So this is the transmission electron microscopy image of silica, and these small particles over the surface is iron oxide, so that we can image using an MRI because it's a contrast agent. Now we also wanted to image it using fluorescent microscopy. So what we did is that we doped a dye inside the particle, then we developed these particles, so that this is the electron microscopy image, and this is the fluorescent nanosilica nanoparticle. We can image it using a fluorescent microscope. Okay, so in both two types of imageable nanoparticles we have developed, one for MRI, one for fluorescent microscopy. Now we took this iron oxide doped silica nanoparticle, we injected it in the mice, put it in the animal MRI, we took out the image at different time points. Okay, so this is iron oxide silica nanoparticle, and this is the cross section at a different time points. As you can see, this is just before injection. Now as the time increase, this area, this is nothing but spleen, it is getting darkened. The contrast agent I used in this work was T2 contrast agent, which is dark in color. Okay, now this is the enlarged image of that spleen. This is the spleen. You look at here, this untreated before injection, and after injection this is getting darkened. That means this particle is slowly getting accumulated in that splenic region due to the presence of iron oxide, and that means silica is getting into the erection. Now, so that is the in MRI data. Now what we did is after 24 hours, we euthanized the mouse and took out the spleen. This is how it looks like our slippers. Okay, the spleen looks like this. See the architecture actually. So this is before injection, the control spleen, before untreated one, and this is the treated one. So you see that this area is getting darkened, and this darkening is mainly in this white pulp region in the spleen. There is in red pulp and white pulp is there in spleen, and the interface of that red pulp and white pulp contains a lot of macrophages. So this interface is getting darkened, which is again an indication that this particle is exactly accumulating into those splenic area. Okay, macrophages of spleen. So this is very important in the case of tolerance induction. I told you, if you can deliver the autoantigen into the lymph node organ macrophages, we can induce this phenomenon tolerance. Now we also checked it through the fluorescent microscopy. We took out the fluorescent silica nanoparticle, we injected into the mice, then we took out the spleen and did an immunofluorescent assay. The macrophages of this region has this antibody called as Marco antibodies. We stained it using that, and our particle was green in color. The macrophages are red in color, therefore stain. And when this red and green converts, it will become yellow. That yellow indicates the particle has accumulated into that specific areas of macrophages in the spleen. So there, which is very important in the induction of this phenomenon called as tolerance. Now you look at this image. This is an electron microscopy of macrophage. Our raw macrophages. This is an electron microscopy of macrophages incubated with a nanoparticle. You see this enlarged image. Lot of engulfing of nanoparticles by these macrophages. You can see macrophage and extensive phagocytosis. You can see, which is not seen with other nanoparticles. So that is the advantage of the engineered silicon nanoparticle. We have got an extremely good accumulation inside these macro. This is a single macrophage cell which has taken up all these silicon nanoparticles. Now we wanted to test it in the animal model. Now in the animal model, I told you the gold standard model of MS is called as EAE. So we induce the disease using the same antigen. Okay, the drug or the compound what we have taken is MOG, and we induce the disease using the same MOG autoantigen. We use some adjuvants to activate the immune system in the mice. We also use this toxin, pertussis toxin, that is used to breach the blood-brain barrier, because MS is a neurodegenerative disorder. So you need to breach the blood-brain barrier. So you use a toxin. So along with this autoantigen, with adjuvants and toxin, we induce this disease by subcut injection. Now later on, after a particular day, 14th day, the animal will end up in severe hind limb paralysis. So this is the disease scoring scale. 0 means normal, 1 means limb tail, 2 means hind limb weakness, 3 means hind limb paralysis. When it reaches 4, that is 4 limb weakness, you need to euthanize the animal according to the IAC. Okay. Now we started off giving 2 doses of injection on before inducing the disease as a kind of a vaccination. We gave it on 7 days before and 1 day before inducing the disease. On day 0, we induced the disease, and we observed the animals for 35 days. And this is the graph you can see. This red graph is nothing but the Moog conjugated silica nanoparticle, the autoantigen conjugated silica nanoparticle, whereas this black and this orange is the control one. So you can see there was significant decrease in the clinical score. Normally the mouse will end up in hind limb paralysis, severe hind limb paralysis. Now the disease has been reduced to 1. So that indicates that the particles that is accumulated in this macrophages of autogenic lymphoid organ is helping in reducing the disease actually. Now this data, this is a comparison of bare Moog, that is just the autoantigen, and this one is the red one is the autoantigen conjugated silica nanoparticle. We have given the bare autoantigen even up to 800 microgram. There was no decrease in the disease. Whereas you will see the nano conjugated autoantigen or the silica conjugated with the Moog with just 50 microgram, it was significantly decreasing the clinical score. This indicates the importance of our nano carrier, which is getting into those lymphoid organs. But whereas if you simply give this autoantigen, it is not getting into the lymphoid organ. So that is the advantage of this nano conjugated or the macrophage specific nanoparticle. Now we took out this spinal cord and took out this we did the histopathology. We can see that in the treated mouse there was significant decrease in the inflammation, demyelination, axonal damage. We confirmed the clinical score was matching with this histopathology, which is very important in the actual scenario. Now we also did a proliferation assay. We took out the spleen and tried to induce the treated mouse spleen as well as the control spleen with the different concentrations of Moog. So normally in the control one, there was severe proliferation, but in the red one, the treated one, there was significant decrease in the autoreactive lymphocytes, the autoreactive T cells. So the proliferation was downregulated, so which is again correlating with the clinical score. Now this is very important part. I told you to bring down the immune system or put an immune suppression, you need to downregulate the pro-inflammatory cytokines. So IL-17 and IL-2, they are considered to be pro-inflammatory cytokines. They will activate the immune system. But you see in the treated mouse, it was significantly decreasing IL-17 and it was also decreasing IL-2. These are some two important cytokines which increases the disease. So we were from the treated groups, we were able to see significant decrease in that cytokines. Now the data what we have seen was with the 200 nanometer particles. We have engineered the particles and the approximate size was 200. We were quite interested to see whether if we increase the size of the nanoparticle, whether it will have a better effect. Okay, so we prepared this 500 nanometer particle. Same characterization we was done. DLS was taken, electron microscopy was taken. This is the image of a 500 sized nanoparticle. Again, spherical in shape, monodispersed, and all that. Now once it is done, we conjugated the autoantigen over to the surface of 500 nanometer particle. We tested it in the animal model. Our purpose was to see whether 200 has the better effect or 500 nanometer better effect. So we compared that this is a size range 200 versus 500. We didn't see much difference. Whether it's 200 or 500 in decreasing the suppression of the disease, we didn't see much difference with 200 versus 500. Probably other materials may show a 300 nanometer difference will create a difference, but with respect to our engineered particles, we didn't see a difference between 200 and 500. Probably we are trying with a higher size like 1000 nanometer. We are doing that studies actually. Maybe that time it may create some difference, but here with these two size, there was no difference. Now, when it comes to so far, I have shown you the data with the silicon nanoparticles, which was a kind of a solid nanoparticle. But when it comes to the translational point of view, the first question is that whether your nanoparticle or nanoformulation is biodegradable. Of course, it is biocompatible, no doubt in that. Whether it's biodegradable or will it create toxicity. So the earlier silica what I have shown was not biodegradable, they are very solid. So our plan was to make it biodegradable. One method to make it biodegradable is that you make it porous. You create pores inside the particle so that it will crumble or it will degrade. Okay, so we tried to create that pores inside the nanoparticle, but our attempts was not successful. The particles were not polydispersed; they were accumulated or they were joined together. So we optimized our chemistry. Later on, we were able to get monodispersed particles with this porosity. This is like a hexagonal comb, honeybee comb. The approximate pore size is just 2 nanometer. But when we checked this, these are very uniform porosity, but the size is very less. The loading of the proteins will be very less. So we again modified whether we can make the porosity into different or higher porosity. So finally we optimized and finally came up with a successful or our planned nanomaterial. This is high porosity, approximately 10 nanometer in pore size. And you can see this small white things inside. These are nothing but pores. And this is highly biodegradable also. So this is a comparison image. The earlier data what I shown was with solid nanoparticle. Now you see this image, the contrast. This is a highly porous stuff and this is highly biodegradable. This is not biodegradable. Okay. Now once it is done, we conjugated this MOG autoantigen onto this. Here the advantage is that compared to other than the outside, we can load this autoantigen inside the particles. The loading efficiency will be very high. Now this is the degradation studies. Within 48 hours we started finding the degradation of this synthesized particle. By 72 hours the inside things are completely degraded. By 8th day and 4th day they started degrading completely. So and one more advantage, as I told, the loading was pretty much high. We were able to get 50% loading efficiency with this material compared to the solid particles. Okay, so that was the advantage of creating the high pores inside the particle. Now we tested it in the animal model again. This time we given after the inducing the disease. On day 0 we induced the disease. On day 14, where the animal will look like this severe hind limb paralysis, the hind limb will be in a paralysis stage, we gave a single shot of injection of the porous nanoparticle with the MOG autoantigen on day 14, and we observed the animals up to 60 days. So this is the control one, blue one. You see the red line, which is the treated one. And following the injection, there is a reversal of the clinical symptom from 3 to 1. The hind limb was getting all right. Now we observed up to 60 days, there was no relapse. There was none of the mouse got any relapse. And by 30 or 40 days, the tail tone also was regained, which indicates the advantage of this porous system compared to the dense system. That too with a single dose of injection. Now we again did the histopathology. We were able to see decreased inflammation. Now we were quite excited to see how the regulatory T cells looks like, because I told you up to 60 days there was no relapse of the disease. That means regulatory T cells would be playing a major role, because regulatory T cells helps to maintain tolerance for a long term. Now you know that normal in our body will be in a balance, inflammatory, pro-inflammatory and anti-inflammatory in a balance. This is mainly mediated by this regulatory T cells. Now we checked this, we wanted to check this regulatory T cells. We took out the spleen and lymph node from the treated group, and we tried to see how this regulatory T cells context is that. You can see the red line where the treated one, in the treated one, the regulatory T cells was upregulating, and that was the reason probably up to 60 days we didn't see any relapse. So regulatory T cells plays a huge role in tolerance and for maintaining the tolerance for a long term. Now this part was work was filed for a patent and published in 2 journals. Now when it again we asked this question, right? So far the data what you have seen was with the IV injection, IV formulation. Okay. Now we were quite interested to see whether the system works through oral route or not. Okay. So because whenever it comes to a translational point of view, always oral will be more welcoming. The regulatory constraints will be very less. So with my PG students, what we did is that we developed the same formulation. We this time we gave to see whether when you are giving through the oral route, how the accumulation in the lymph node organ. So one of the major lymph node organ when you are giving through the oral route will be the Peyer's patch, the small intestine, the gut associated lymphoid tissue. So we gave this formulation through the oral route and took out the small intestine, took out the Peyer's patch, and did an electron microscopy to see whether the accumulation of this particle. So this is an untreated group and this is a treated group. So we were able to see that our particles are accumulating into those Peyer's patch, which is very important for inducing oral tolerance. So that is the image of that. Now this uptake by Peyer's patch is very much significant in the induction of tolerance. Now once we have seen that it is accumulated in the Peyer's patch, we gave the treatment, this time through the oral route. Earlier it was IV route, this time through the oral route. Same way, on day 14 we given the treatment, on day 0 we induced the disease, on 14th day we given this formulation. Similar to previous study, we observed the animals for 60 days. Now you see the red line was the IV treated one, blue was the control, this green is the orally treated. Although it took some time to reduce the disease, it was having after 25th day, this oral route was having a better efficacy than the IV route. That too, again, a single shot of injection, but the quantity of peptide we used was 4 times more, 100 microgram, because you know that some of the peptide would be degraded definitely in the GIT. Okay, the autoantigen or the MOG on the surface will be definitely degraded in the GIT. Only the MOG autoantigen which is present inside the pores of the nanoparticle will be remaining. So we used a bit higher dose of this autoantigen, that too in microgram scale only. And we were able to see, compared to IV, oral was having even better efficacy. So that was eye-opening to us actually, because although we did the proof of study with the IV route, this oral data was actually much promising, and we really wanted to take it forward actually. Now this is those animals with a severe hind limb paralysis. This animal is the EA mouse, and this is that orally treated mouse. BD on the plate. Just play. Yeah. Just to play that. So this is that mouse. You see, they are having severe hind limb paralysis. This walks like this. Clinical score of 3. Upper. See, the same mouse at the end of the treatment was regaining the tail tone, and the hind limb also was getting okay. No, so the conclusion is that we can engineer these biodegradable nanoparticles. We can load proteins, autoantigens, and we can specifically target it into those macrophages, whether it's spleen through the IV route or Peyer's patch through the oral route, and we can induce this phenomenon called as tolerance, which is very much important in regulating autoimmune disease like multiple sclerosis. Now we are this platform technology can be extended to other autoimmune diseases, because you know that compared to MS, rheumatoid arthritis is very common in Kerala, very common in India. So that would be having a better effect, a better edge over even. So similarly, same way, we recently received a grant from ANRF, ARG grant. This was for rheumatoid arthritis. The same technology proposal we applied, and this was recently funded. Hopefully we are expecting the funding to be getting in March, at the end of the March. So there is an open position of junior research fellow. Some of your friends, if you are interested to work in this area, welcoming, and you can send a mail to this mail ID, your CV and all those things. So this is for rheumatoid arthritis, okay, which will be more relevant in the context of Kerala scenario or the Indian scenario. Now, apart from this, whatever thing I was showing with the autoimmunity, now we are also working in collaboration with other areas. This Indo-Switzerland project, where my whole objective was to develop. They asked this question: can I develop a nanoparticle with an immunostimulant action? So this is the same silica nanoparticle what I have shown, but here I loaded this aluminum, which is an adjuvant. So also we have loaded this manganese. So when you are loading aluminum and manganese, that is you can see this a shell kind of thing and a core kind of thing. That is the difference of this aluminum. Now when you are loading with aluminum and manganese, we are able to see that the immune system was activating. That means the same material we can use in the two opposite contexts, cancer as well as autoimmune. It's just a matter of engineering or adding the right material, right engineering that will help. So this is actually working well actually. Now this is another project where you have heard about the CRISPR-Cas, the gene editing tool. To this is our silica material, highly porous material. We can load this gene editing tool into this. We can use in this this CRISPR-Cas therapy based on the gene editing tool. Again, that is another thing. Now we also made, as I told, we also made this magnetic nanoparticle. We doped iron oxide onto this thing. It made it magnetic, and that can attract this circulating tumor cells, which could be detected by Raman spectroscopy. This is another work which is going on actually. Now we also we are also looking into the tissue regeneration with the same material. Everything, the porous material, just changing the engineering, the chemistry of that, and we were able to see that this irisin is nothing but a bond regenerating material. But when you are giving bare irisin, the effect was not good. But when you are loading this irisin into this silicon nanoparticle, that porous material, that was having a better tissue regeneration, and we are this resulted in a publication in Precision Nanomedicine. Now this is one of my own students' work actually, which is very close to my heart. Now earlier what you was seeing was the spherical nanoparticles, the round particles. Now I am quite interested to see I also shown that the 200 and 500 was not having much difference. The size was not having much difference. Now I just wanted to see whether the shape will create a difference. So this is that see bacteria looking like bacteria shaped like bacilli shaped silica nanoparticles only. So this looks like a road, and this is just like almost 1 micrometer in length and 200 nanometer in width. We can increase the size also, just like you can make it like a needle like this. You can make it like a needle, or just optimizing the chemistry, you can make it into a different shape. And this will have a different immune activation, maybe immune suppression, or sorry, maybe a higher immune activation compared to the spherical particles. We don't know. Still, we are under investigation that. So again, I'm telling the shape can create a difference, or you add an adjuvant to that, it can create a difference. So these are some of the works, and thanks to our collaborator from Amrita and Monash University, Australia. And once again, I thank MG University and the organizers of this 2-day seminar program for giving me an opportunity to present my research work. Open for questions. Thank you.
Thank you, sir, for that insightful, inspiring session. Now the panel is open for discussion. You can ask your questions.
Thank you, sir. Thank you for the insightful talk. I have few queries. So about the nanoparticle size, you compared 200 nanometer with 500 nanometer. So why you specific with this 200 and
Exactly. That's
why
a
you
good.
cannot come
Yes.
down
Yeah.
like 20, 50?
That's a correct question. So I think I missed that intro part due to the limitation of time. So normally you take a general information about nanoparticle is that I told 1 to 1000 is considered to be a nanoparticle scale. So when it's too small, like 10 nanometer, 20 nanometer, it will be rapidly eliminated to the kidney. Now, when you really wanted to target a drug, mostly the nanoparticle used will be less than 100, maybe 50, 80 nanometer, 90 nanometer, 100 nanometer. When it goes above 100 to 500 range, that is the limitation of nanotherapy. They will be taken up by the phagocytic system. Okay, that is why in cancer therapy or drug delivery, mostly the people will use nanoparticle less than 100. But when it goes above 100 up to 500, the chance of uptake by the phagocytic system or macrophage will be high. So that's why to kickstart the work, we started off with the 200 nanometer size. And 200 was already, I told you, it was having, compared to polymeric 200 nanometer particle, this engineered silicon nanoparticle was having more affinity towards this macrophages. And we wanted that. In autoimmune condition, we wanted that particle to be uptaken by the macrophages. So that's why we kickstarted with the 200 nanometer particle. Then we wanted to see, our hypothesis was that when it goes to higher size, it will be more uptaken. So that's why we used 500 actually. But we didn't see, in the actual disease scenario, we didn't see much difference. That 300 nanometer difference was not really significant to show a difference. Maybe 800 or 900 or 1000 would be enough. But the problem is that when you really go to higher size, the toxicity problem can come. So this is an ideal range to play with autoimmune disease.
One more thing about the porous silica.
Yes.
So when you are reproducing the results, the porosity, could you control the porosity?
Exactly.
And also one more thing, how could you control delivering of whatever it is possible?
Yes. So I shown you two porous particle. One was that honeycomb shaped, very hexagonal, small pore size actually. So one disadvantage is that you cannot load a lot of proteins into that. The pore size is small. But when it was a higher size or a disordered porosity, it's just like you take a football, you just insert with pinch it with a knife. So the porous, it will not be uniform. So that interaction of the protein and that porous nature inside was better actually. Okay, so that is the first thing. Now, how to control the release? That again yet to be studied. We don't know whether it's a bulk release or a sustained release. But one thing we know, at the third day, after 48 hours, this particle started degrading. So before itself, the release would be starting. We didn't went into the release kinetic studies so far, but we are expecting within maybe a bulk release or a sustained release might have happened. At this point, I cannot answer to this question.
Because we read many articles like a controlled delivery.
Yes.
Some drugs, the porous materials are good for controlled delivery.
Controlled
But
delivery.
we were studied about the kinetics of the controlled delivery,
Yes.
how it happens after a time t or
Yeah.
something.
I think the first one particle, that hexagonal shaped porous particle will be better in that control release because it will not be fastly released. This is a disordered porosity, maybe fastly it will be coming. However, with a single, from the experiment point of view, we didn't see any relapse up to 60 days. And this was working with other research areas also. Like I told you, the irisin protein, bond regeneration, it is working actually. So maybe further we need to investigate, but at this point I can tell that's we are not.
Thank you. Thank you.
Is there any other questions?
Dr. Prashant, it was a very nice insightful work. We would also be looking forward what is the possibilities or to what extent the clinical trials something is recommended in this direction in different parts of the world.
Yeah. So I told you in autoimmune disease there will be a victim autoantigen. Okay, in MS it was that myelin proteins present in the myelin sheath in the spinal cord. So we have taken that one protein out of the 2 or 3 proteins. We have taken only one protein. So the next plan will be use the entire protein, entire myelin. You take out the myelin sheath, get the entire protein, then put into this thing. Here we have used only one sequence of that protein actually, or that peptide. So in actual scenario, we don't know because whether the immune system is going attacking only that MOG and auto, we don't know. It will be definitely attacking MBP. It will be definitely attacking other proteins also. So the better translational point will be use a cocktail of that protein, load it into these particles and try the experiment. That actually we have already started it actually. So that is the actual scenario.
Another doubt, what is about the, now when we work with silica and all that, we always speak about the toxicity.
Toxicity, perfect.
Toxicity. Now coming to silica nanoparticles, the ones which we are creating.
Yes,
now,
that's a good question.
What about the toxicity of this?
Yeah. So silica normally it's a well explored material. Only thing you need to engineer it in the right way for your desired purpose. It is having a GRAS status in pharmaceutical industry, generally regarded as safe. It's used as an excipient in tablets or formulation. And silica is extensively studied even up to mg scale, 500 mg per k, like that in the animal selection. Here we are just using 100 microgram. They have extensively studied up to that high mg scale up to in the mice and all that. It's having not problem. And we are using a 100 microgram, which is 1,000 folds less than that. The second thing is that the first particle what I shown was solid, which I will not welcome in the actual translational point of view. The second particle, which is a highly biodegradable stuff, is actually degrading and it will be converted to silicic acid. It's a water soluble stuff. It can be eliminated from the body. So in that point, this is a completely safe stuff actually. And the dose is very, very less. And people have studied even 1,000 times more higher dose compared to in other studies. That is.
Is there any other?
Sir.
Very nice work.
Thanks.
So I was wondering, I was always thinking that MS is not an Indian disease, but you said that MS is there, like incidence of MS.
Yeah, so earlier it was a Western thing actually because because this animal model when we try to develop, it's a big challenge.
Right.
In Indian breeding condition, it's very difficult to develop that animal model.
Right.
But where if you go to Australia or US and try to develop this animal model, 50 out of 50 animal will develop the disease. Where in Indian scenario it will be less. So that environment plays a huge role. So our climate was actually good enough to keep MS away. But as I told, the recent strategy or the hospital scenario, every week 3 to 4 patients at that particular hospital which I mentioned, people were coming, that to young adults actually. It's popping up actually.
So
It's increasing.
young females are coming?
Especially a female, because there is a genetic component actually. 3:1, female 3, male 1. 3:1 ratio is also there. For many autoimmune disease, it's considered to be more for female actually. And MS is not an exception, and the ratio is also there. So it's I personally know many people, even my colleagues actually getting into clinic with small symptoms. So that's why initially when you take this treatment the right way or whatever thing, it will not make the patient get into that wheelchair or whatever thing actually. So that is the thing. Current treatment is immunosuppressants, which is not the right way to.
I'd like to know more about the patients. Like since you are in hospital, so like what kind? Because MS is of different types, like
Yes.
RRMS and this primary progressive.
Exactly.
What kind of disease we have in here?
Commonly RRMS, relapsing. See, that is why once the patient is diagnosed with MS, he or she will be having a peak symptom. After 2 months, it will be normal actually. Again, after 6 months, it will pop up. So that is a relapsing, remitting model actually.
So that also you see here.
That is the maximum.
Okay.
Progressive is like that. The disease keeps on increasing without any relapse. But so that is
The patient ultimately die, actually.
that is right.
I have another technical question about your work actually. So these silica particles along with that MOG 37 to 52, So that is going inside the, going inside the spleen. Right?
Spleen or lipase patch or lymph node, whatever thing.
Yeah, the lymphoid organ. So how is it affecting the brain? Because ultimately MS is a brain disorder.
Okay.
So how it is affecting the brain?
So although MS is a neurodegenerative disorder, which is happening, all those chaotic things are happening in the brain.
Yeah, it's
The
a
initial,
brain disorder.
yeah, the initial triggering, everything happens in the periphery, because I think the first slide I shown actually. They will cross the BBB, all this infiltrating immune, activated immune cell, they will cross the BBB and will reach the brain actually. So we are not trying to deliver the drug into brain. We are just concentrating on the that is why that word peripheral immune tolerance. We are trying to do it in the local periphery. So that further infiltration or activation will not be there actually.
So are these, because I work on brain actually, so these this nanoparticles, can this cross the blood-brain barrier and go to the brain?
As such, this will not be able to think, because right now the engineered thing is kind of hydrophilic. Okay, it will not be able to cross actually, but we can modify the surface chemistry and we can get it made. But for our purpose, it is not really required actually.
Yeah, I understand.
We wanted that lymph node, okay, either spleen or Peyer's patch or lymph node. That is our purpose actually. But still, the composition could be modified to cross the BBB.
Very nice, sir. Thank you. We'll talk about it.
Sure, thanks.
It was a very wonderful session. And I have two questions. First one is that how did you manage to optimize the size of the nanoparticles, 200
Yes.
nanometer and 500 nanometer? And second one, you said that cancer and these type of treatment methods are actually like playing chess, and you can get another move from the body. So are you expecting any challenges, and how are you thinking about facing it?
Yeah, we'll take up the second question first. Definitely, see, the scenario I told is in cancer. Whatever this is not my word actually or sentence, actually this is told by Nobel laureates actually. Whatever move we take against cancer, the cancer will have a counter move, just by changing the mutation or changing the mode or surface things or whatever thing. So similarly, in the case of autoimmunity, it could be there, but unlike cancer, we don't expect much in the case of autoimmunity. Because and one more thing is that many things in autoimmune disease is still unexplored. Okay, so that is also, we don't know, in future sometimes it can change, because right now the literature or whatever data what we have, we say that this, for example in MS, it goes and attacks the myelin antigen. Still we don't know whether it's the MOG or PLP or the other one is which is the most attacked protein. The data is still not there actually. That's why I told you, in actual scenario we need to take the entire protein from the myelin, or we need to take that entire protein, put it into the nanoparticle and check it. That is the best thing. And coming to your first question, how to adjust the size. So that was actually a tricky, it almost took more than 6 months to 1 year to adjust the composition, size and all that. It depends on a lot of factors. The precursor what you take, the surfactant what you take, even the drop rate. I'm just telling my practical experience. See, if you're adding something from this height and your beaker is here and you're adding through the pipette at this height, you try it like this or you try it like this, different context is there. The rotation, RPM, all these are, you need to play around it by changing the catalyst. For example, you use the base ammonium hydroxide, sometimes NaOH will be better actually. So that chemistry, even I'm telling this height matters a lot. So even a lot of things. See, you are dealing with such a small size, 200 versus 500. We cannot even distinguish. That is, although there is a 300 nanometer difference, it's very small. We cannot detect actually. So to adjust that chemistry, it's really everything. Your hands should be perfect, or reproducibility should be there actually. Or you need to develop a technique where you keep a distance or whatever, or rotation, RPM, or like that. You need to really optimize. Note down all those things, trial and errors. Then you will get an algorithm to get it to the final one. That I really expect. Because I told you, 6 months it took. My guide actually told we tried for 500. We are not getting. He told to drop that. I told give me 2 more chances, because I know I was about to reach that by like an algorithm. Like you change the ratio, you know that you were able to create 300, you were able to create 400. Now just 1 or 2 steps away to create 5. So I just told him, give me just 2 more chances and record it actually. So I'm telling, note it down, everything, whatever you do in the lab, or not only this, whatever you do, just note it down. It's very important.
cell attack our own cells? What is actually
Yeah.
the core cause behind autoimmune?
Till now, the exact reason is we don't know. Like how that immune system actually getting active. I told you, we all take this food antigens, like non-veg, veg, but most of us are not getting into autoimmunity. Why that getting into thing, or what is the exact reason? Still is unknown. But certain studies tell that certain virus or some virus thing which we entered, or that is remaining as a particle there, could trigger the peripheral immune system. They can activate that. They can further close those things, BBB or whatever. It can activate the system and create a pro-inflammatory cytokines and that. Exact reason. So even this differentiation with the female to male, I told 3:1. Again, hormonal studies are there and all that, but still the exact reason not explored actually.
Doctor, I would also like to know an opinion on why, like now, you had told 18 to 20, especially females, are getting multiple sclerosis,
Yes.
and it was not present in so much frequency in the Indian scenario. It was more in the Western
scenario.
So, could you pinpoint some reason probably which is leading into this change?
Yeah, one thing is that see, there is a study based on vitamin D. Like we are living in a tropical climate region where we have enough sunlight and all that. Whereas the Western, they have 6 months like not having. So that is one of the literature says that actually that vitamin D could be one reason that the Indian scenario, tropical countries have lesser thing where compared to the West. Now in this male to female that thing you have told, hormonal things has been demonstrated, but still that exact reason is not there actually. Well, most of the thing, the sex hormones based things, they have tried to correlate with the more incidence on females compared to the males. So that is still not an exact reason we could say. And environment, I told you this when we induce this disease, some of the I got an opportunity to visit Australia and do this animal studies actually. So in Australia, when I went there and tried to induce disease in 50 animals, on 14th day, all 50 animals disease was induced. Same thing I did it in here. 50 animals you try to induce, 15 or 10 to 15. You really need the breeding condition, the environment, all the animal house things are very important. That even that humidity matter or this dampness on the bed where the animal lie. We have changed the husk to corn cob. There was a difference. So a lot of things we have to optimize. The environment really plays actually like that. Same way it could be related to humans also.